Vibration-cued hatching is widespread, diverse, and important for survival in many animals. From insects and flatworms to frogs and turtles, embryos of all sorts use disturbance cues to inform their hatching timing. Yet the mechanisms mediating vibration sensing in ovo are largely unknown. We know most about vibration-cued hatching in red-eyed tree frogs, which lay their eggs on plants overhanging rainforest ponds. Usually within 7 days, embryos hatch into the water below and develop as tadpoles. But they can hatch prematurely to escape from egg predators, cued by vibrations from attacks. This escape hatching response changes drastically over development. By 3 days, embryos hatch in response to hypoxia in flooding. Predator-induced hatching starts about a day later, meaning there's a period of development where embryos being attacked by snakes are clearly able to hatch, but don't and just get eaten. Since hatching ability does not mediate the onset of predator-induced hatching, perhaps sensory ability does. To test this hypothesis, we examined the development of two well-known vertebrate motion detectors, ears and the lateral line system. Both depend critically on mechanosensory hair cells. To see if either of these mechanisms enable embryos to sense predator attacks, we measured hatching responses in embryos with both with and without ear function, with and without lateral line function. To measure ear function, we used a behavioral indicator called the vestibular ocular reflex. To calculate reflex strength, we used a tadpole rotator that holds a newly hatched tadpole in a small tube so we can take portraits of them in different positions and measure changes in eye angle relative to body rotation. The amplitude of the resulting curve indicates the magnitude of ear function in the developing embryo. Here we show embryos before and after ear function. To measure lateral line function, we labeled hair cell bundles using a fluorophore called diasp, which temporarily dyes hair cells orange, showing functional neuromasts in live embryos. We treated embryos with gentamicin to temporarily ablate lateral line function. To induce hatching, we simulated snake attack by jiggling eggs with a blunt probe in a standardized pattern, presenting a complex physical disturbance. We did this to 4 different groups of embryos before and just after the onset of ear function, after vestibular function was well established, and close to spontaneous hatching. And in each of the 4 groups, we tested embryos both with and without lateral lines. This shows hatching responses before and just after the onset of ear function. In both cases, lateral line knockout in red reduced hatching. These embryos overlap in age, but only the green group has functioning ears. Ear function seems to increase overall hatching, but even before it begins, lateral line function alone enables some attack-induced hatching. The hatching response increases developmentally. Here, lateral line ablation still reduces hatching. But our oldest embryos all hatched, so we couldn't detect a gentamicin treatment effect. We examined latency to hatch as a potentially more sensitive indicator and found that even these older embryos with well-developed ears still use lateral line input to accelerate their response. But not all embryos receive complex physical disturbance cues in snake attacks. Some receive only egg motion cues, transmitted through the clutch. To assess if egg motion alone may stimulate the lateral line system, we used vibration playbacks to shake treated and untreated embryos held in custom-made egg trays. We found no effect of neuromast ablation on embryos' hatching response or timing of hatching in motion-only vibration playbacks. In summary, it appears that in attacked egg clutches, some embryos receive motion cues via ear stimulation and the lateral line makes no contribution to hatching, whereas other embryos receive disturbance cues via both the ear and the lateral line. Since lateral line input plays a role in the perception of some disturbance cues, we examined how the lateral line system develops across and beyond the onset of the hatching response. We used diasps to label neuromasts and photographed individuals in frontal and dorsal views to count them. Neuromast numbers in all 7 lines increased with development. This ontogeny aligns with the known onset of attack-induced hatching. From when it starts to when it becomes consistent, the total number of neuromasts more than doubles. Neuromast number predicts hatching in response to egg jiggling. Below a threshold number of neuromasts, no embryos hatched, whereas embryos with more neuromasts often hatched, suggesting lateral line development may play a key role in the perception of predator cues. The number of neuromasts at hatching varies among species. Some fishes hatch with just 2 neuromasts, and early larvae simply float in the water column. Others migrate downstream upon hatching and do so with 40 well-developed neuromasts. Red-eyed tree frog embryos hatch with many more neuromasts than have been found in any other species. Egg predation might be a selective force favoring earlier or greater development of embryonic sensory systems. Studying how sensory development enables embryo escape behavior reveals the key role of environmental information even at the earliest life stages. Thank you so much to my colleagues, collaborators, and funding sources, and to you all for sharing the virtual table with me today.